Okay, everyone, I'm going to get started. Thank you for coming to today's seminar. The first thing we're going to do is hand out the awards from the colloquium last week. This isn't a surprise because it went out in email because I know a lot of people aren't here. But this year, the awards committee decided that they wanted to award master's students and not just PhDs, which is great because Jemana won the presentation award for Outstanding Master's Student. Congratulations. And second place, I guess it would be overall, is Jay. Congratulations.
Thank you.
And first place is Grayson. Congratulations.
Thank you.
Okay, so today I didn't prepare an introduction. That's okay. I think I'm going to try and do this from memory. Today it's like my pleasure because I get to introduce, and what's really cool is if any of you become faculty, you get to invite your friends to come to seminars and then an excuse to hang out with them. So Pierre's one of my closest friends from graduate school. We started at the same time, our PhDs together. When I first met Pierre, he was an introverted student from Los Angeles, and pretty quickly that turned around after an ice cream social event. Pierre was in the honeybee lab at A&M. He studies nutritional ecology. He focused a lot on pollen, so what I know about all of Pierre's talks is that we'll see some beautiful pictures of pollen. I hope that's the same today. Is it?
A little
Surprise?
bit.
A little bit.
Yeah.
We used to see a lot more. He's changed his ways. Comes from Los Angeles. He did his undergrad in UC San Diego. Did his PhD at A&M, Texas A&M. Woo. And now he's a USDA researcher in Mississippi. So he's going to be telling us about the work he's been doing there. Did I forget anything? And I didn't say anything embarrassing. So.
Yeah.
Which I could have.
Which you could have. I would have got you back for sure. But anyways, so hi. Thanks for having me and inviting me, Chloe. Thanks for the department for hosting, and it's been lovely meeting everyone so far. So I just want to start off by just briefly introducing the research unit that I'm part of. It's relatively new. I've been there for about 3 years. I'm one of the first 5 scientists that were hired there to help start this unit. But we have our research leader, Dr. Rathi Sosardi. She has a background in plant biology and also works on pollinator health. Dr. Kathleen Paris works on the native bee side of things. She is more of a taxonomist. I am a research ecologist, and my interests, you'll see soon enough, are in nutrition ecology. And Dr. Yu Cheng Zhu is a toxicologist in our unit. Dr. Zhang is a research chemist, and Dr. Cannon is our ag engineer. And then my research program within this whole unit can be summarized in 3 main areas. So first is to understand basic bee nutrition, and there's still a lot that we don't know about general honeybee biology and nutrition ecology, so we are still working on this area of research. And then we are expanding on that and adding interactions. So how can we use nutrition to improve pollinator health, especially in the agricultural settings when there are lots of pesticides being applied? So how can nutrition and pesticides interact with one another so that we can figure out ways we can reduce susceptibility, bee susceptibility to pesticides and other toxins out there in the environment. And last but not least, we are looking at ways we can optimize agriculture management practices, and I will get into that in more detail towards the end of my talk. But before I move forward, I want to first acknowledge the lab members that have been part of my lab over the past 3 years. None of this work would have been possible without them, especially Giovanni, who is my technician, and he's basically our lab manager who runs the lab. So I like starting off with this little sketch. There are just many different factors that affect bee health: pesticides, pest parasites, landscape change, and poor nutrition. A lot of my work is focused on the nutrition side of things, but in this agricultural system, everything is kind of exacerbated because you have intensive ag systems where you have monocultures of resources, so bees don't have much of a choice in what they forage for. And you also have pesticides where they're being applied heavily into the environment. So over the course of my career, I've been able to focus my studies on just everything about bee nutrition. So I've worked with nectar, pollen, and water foraging. So here are the pollen images Chloe was talking about. But I think this is the only slide that I have pictures of pollen. But bulk of my research is on pollen and bee foraging. So here is an example of some of the tools that we use. We use palynology, we look at the pollen and we identify them, and we look at what bees are collecting. We've also done some work on the types of water that honeybees like collecting, and it's heavily influenced by the minor salt concentrations in there. So Mike, you were talking about beekeepers or people putting out water out in the environment, right? Well, some of that is attributed to some of the knowledge that's out there that bees need water in the environment, and they use the salts in the water to complement their dietary nutrition. And then we've also done some work with the nectar and the honey, and this is just like a map of the different types of honey and the pollen types that bees will collect across the different ecoregions of Texas. But while I was doing all this honeybee foraging research and looking at what they prefer, I had a lot of questions from beekeepers whenever I was presenting to them and my other stakeholders. What's like why are they going to certain plants? Is there like a preference for one plant over another? And for me, I thought there was a nutritional basis for foraging. So we decided to pursue this question based on the literature that we've looked at, and we realized that there are still knowledge gaps in this area. So the model that I use to pursue this question is the geometric framework for nutrition. It's basically a modeling approach where we can explore how animals can solve the problem of having multiple nutrients at one time in a variable environment. So in this example here, you have an organism that has two choices of diets, one that's high in protein and low in carbohydrates, and one that's high in carbohydrates and low in protein. And this is the optimal intake target that the organism needs to reach in order to maximize its fitness at a particular time. But there's no diet that will take that organism to that specific intake target. So what this organism can do is eat both of these foods to complement its diet in order to reach this optimal intake target for its nutritional needs. And this is pretty commonly used, and it's being used more and more now to determine macronutrient ratios for different animals that can affect survival and physiology. So I love using this example. The New Zealand, I learned it's called kakapo parrot. It's not a kakapo parrot, but this is an endemic parrot to New Zealand area. It's critically endangered. I also love this example because it's also known as the world's fattest parrot. So these conservatories were trying to rear them back up in their conservatories and their zoos, but these birds weren't reproducing, and the people couldn't figure out why that was. They were feeding them the supplementary feed. They brought in two of the scientists, Dave Robbenheimer and Stephen Simpson, who basically pioneered this or started this whole geometric framework for nutrition. And they looked at the natural diet of the parrot, which is a rimu berry, and found that there was a mismatch of the protein to lipid ratio, and the calcium levels were different than what the supplementary feed were giving them. Conservatories were providing them. And calcium is important for these birds in order to develop their eggshells for reproduction. So in 2019, there was this article. I mean, it's not the direct cause, but it was one of the reasons why these parrots have bounced back from being critically endangered in New Zealand. So when we look at honeybees, we see a very similar case. This is a graph where you have time on the x-axis and then the number of adults on the y-axis during the summer months from a study done in the University of Florida. They compared different diets over time on colony population size. This yellow dotted line here are the colonies given natural wildflower pollen. All of these other lines were given an artificial diet of some sort. And this purple line here is genetic control, where they're not giving any type of diet at all. So all this really tells me is that the nutritional basis of these pollen supplements is poorly understood, as Dr. Jerry Wright said, and a more rational approach on honeybee ecology and physiology is needed, because none of these artificial supplements are outperforming really the negative control, and the bees giving natural pollen are still doing much better. So we aim to do this whole nutritional geometry framework on honeybees, but we first wanted to start by looking at some of their favorite foods and their diets, because there's this idea that bees kind of have it figured out and know what they like foraging for. So we took two pollen types, Brassica and Rosa, both of which are favorites of honeybees, and we analyzed its nutritional properties, and we see that they are completely different from one another. These are just elemental compositions of the two pollen types. And you can see the actual numbers here. It's almost completely inverse from each other in terms of these different elements in the pollen. And we found a second thing here during the study. While we were analyzing these pollens, it took like an entire year because we realized these methods were just very difficult to work with pollen. Where pollen is very difficult for these methods to successfully be used on pollen, because pollen contains this outer exoskeleton type thing. It's not like exoskeleton, but it's an exine that protects the inner nutrients of the pollen grains themselves. So when you just send off your pollen samples for analyses, and they analyze the pollen, a lot of times they're not getting what's inside of the pollen grain. So we found that you actually need to disrupt the pollen and crack the pollen open in order to fully access the nutrients in there. So in this Brassica example here, you see that when we disrupt the pollen, we get much higher fatty acid yield, and we also saw that for protein and lipids as well too for Brassica. But we don't see that for Rosa pollen, and so there's a lot of variation with how pollen analysis, and this is a whole paper that I can get into for like a whole other talk, but I'm not going to talk about that too much. So after we determined the protein lipid ratios of these two pollen types, we just present them with a choice test, gave them to colonies that are contained in the nucleus in the tent, so they can't forage in anything else. And we see that bees were constantly visiting Brassica but not Rosa, even though they have a very similar protein to lipid ratio. So we're like wondering why that might be, and so we had some suspicions that the fatty acids might play a larger role in that. So we also wanted to look at this in a more controlled setting with nurse bees, because they're the ones actually consuming the diets. So we created some artificial diets containing proteins, lipids, sugars, and a few other things. I'm not going to get into those details. And we created these nutrient rails of artificial diets that we presented to bees. And we did a cage assay where we gave them a choice or we gave them a no choice, and we measured their physiology afterwards and assessed their health. And with this whole geometric framework applied, this model, we basically found that bees have an overall intake target of about 1.4 to 1 protein to lipid. And I can just walk you through this real quick. This triangle here, this treatment group, where bees given 35-15 and 35 parts protein, 15 parts lipid, and this other group here is giving 25-25. They're given a choice of these two. And so the bees that ate to this point were given this rail and this rail, and they consumed both at a relatively equal amount to get to this point. When they're given a high protein 35-15 to 20-30, they consumed the 35-15 significantly more than the 20-30, but they also landed on this rail. And then when bees were given the 30-20 and 20-30, so these two, they consumed both of those diets, but significantly more of the 30-20 and landed right here. So on average, they had a PL intake of about 1.4 to 1. Now, how does this relate to our current industry? There is a huge mismatch. A lot of the protein supplements that beekeepers have access to from commercial companies are about like 40% protein and about, let's say, 2% lipids. This one's 50% protein, 4% lipids. So it's almost like 10 to 1 protein lipids. So there's a huge mismatch of what's available in the diet industry for honeybees versus what we are seeing. And in this paper by Anthony Votto, they summarized the protein lipid content of all these different pollen types that different bees are collecting, honeybees, bumblebees, and osmia. And honeybees landed right around here, and they estimated that based on the pollen nutrition analyses, honeybees would collect between a 2 to 1 to 1 to 1 protein to lipid ratio in their pollen diets. Now, but what if we introduce a stressor into the system? So the intake target right here is not necessarily static at a given point. And it can actually shift depending on the physiological state of an organism or an animal. So let's say that you are feeling sick, right? A lot of times when I'm sick, I'm going to maybe want to eat chicken noodle soup. You know, you have these kind of tendencies and of foods that you go for whenever you're sick. So that's like the idea of this system here. So, and it's been shown in a few instances here as well. So unparasitized Manduca sexta larvae normally regulate protein and carbohydrate ratio about 1:1. But once they are parasitized, the larvae can completely abandon their strategy and will randomly forage in the environment. Some other fire ants, on the other hand, once they're parasitized with Solenectida invicta virus, they will actually shift their diets as well and collect a completely different ratio. So these stressors can vary from viral mites to pathogens, pesticides, to temperature. And in our case, since I'm in the Mississippi Delta where it's heavy ag, we are looking at pesticides. So one of the first ones we're looking at now is clothianidin, and this is a neonicotinoid that's commonly used for controlling pests in corn, soybeans, cotton, and even Salicto concentrations of these can actually impair B lipid and glycogen levels and learning and memory. So this is a very well-studied pesticide already, which is why we wanted to pursue and continue with this. So we set up these same cage studies, but we added an interactive effect here of the pesticides. So we had one that was giving sucrose, 5 parts per billion and 20 parts per billion clothianidin. And then we had our 30-20 diet that we found that they liked. And then we also had a high protein diet and a high lipid diet. We also had a negative control with no diet and a positive control with pollen across these 3 treatment groups. And we did a Cox proportional hazards model here, and this one, A is our 0 parts per billion, B is our 5, and then C is our 20 parts per billion. And what we basically saw here was that the no diet and the 20-30 diet was consistently, the bees giving those diets consistently died faster. Okay, and then the bees given the pollen 40-10 and 30-20 diet performed relatively similarly. But when they were given the 20 parts per billion, the higher concentration of clothianidin, we actually start seeing the pollen group diverge from the bees given pollen do better than the other groups as well, especially the no diet and the 20-30 group. And we also looked at some other aspects of their physiology and enzymes. So this is just one of the panels of graphs. I'm not going to go into it in super detail, but overall the trend that we saw was pollen increased bee physiology, so hypopharyngeal gland and abdominal protein, and also enzyme activity, so glutathione, GST, esterase, and invertase levels the most. But having the 40-10 and 30-20, the higher protein diets, actually did help them a little bit. So it didn't perform as well as the pollen diets, but it did help them out. And there was no interactive effect of the pesticide or interaction. We also looked at the different families of the P450s and also the vitellogenin gene expression. And one thing that stood out was that the bees given natural pollen consistently upregulated their CYP450 genes, these different groups, compared to, besides CYP9Q2, compared to all the other treatment groups. So what we concluded with that was that there might be other nutrients in the pollen outside of the macronutrients that have more of an influence with immunity compared to the diet, the artificial diets, the macronutrients in there. Vitellogenin was also highest in the pollen bees, but it will also promote it in the bees fed 40-10 and 30-20 diets. So for that study, we saw that nutrition has a direct effect on promoting bee health and longevity. Proteins and lipids do help bee physiology markers, but they don't do much with upregulating the P450 gene expression levels. So the macronutrients alone in these artificial diets weren't the reason or the cause that the P450 levels that are involved with upregulating pesticide, or that are involved with detoxifying pesticide exposure. Yeah. So yeah, the macronutrients alone that we're responsible for upregulating, we're not responsible to upregulating detoxification methods. But instead, other parts of the pollen, and based on the current literature, we suspect that it might be the phytochemicals have a larger role for promoting the immune response. And the reason why I do think it is the phytochemicals, and the phytochemicals have been studied by labs in University of Maryland. But also, for plants, they don't necessarily want to be preyed on by herbivores, so they have these defenses, which are phytochemicals, to deter herbivory on the plants themselves. So these insects would evolve to be able to tolerate these phytochemicals, and these are the plant defenses, so it would upregulate their immune defense genes. So we think that there's something going on here with the bees as well, even though they definitely have a different objective than the herbivores. And the last area that we are really focused on is the overall nutritional landscape. And so here we have a grocery store that's filled, that like the market. For bees, that's like the same thing. So each one of these products will have a different nutrient that you have access to. For bees, all these different plants will have different nutritional resources that they have access for, like shopping for. And this here is a really cool tool called Beescape. You can enter in your location, so I put the department here, and it will generate a little graph or an image or statistics on the nesting availability for wild bees, or the amount of floral resources in an area, or even they have an index for crop insecticide index. So this is from Penn State, and they collaborated and got funded from the USDA and generated this tool where you can enter an address, and they'll give you like an index on how good a place a specific location is for pollinators. But the nutritional landscape can vary in space and time, and especially when you have seasons where there aren't many things blooming. For us, it can look like an empty grocery store whenever a hurricane is coming through, for example. Everyone's just panicking, grabbing everything. For bees, summer months, there's not much blooming out there in the environment at all. And another time this happens is when you have a lot of bees in one specific area at a given time. So when you look at the commercial beekeeping industry, you have 800 or 1,000 or thousands of bee colonies at one space, especially when you have almonds that are about to bloom. Everyone's moving their bee colonies over to California, waiting for the almonds to bloom. But prior to them blooming, you don't really have anything out there for them to forage on. You also kind of have all these bee colonies in one space at a given time, where you kind of exceed the carrying capacity of an environment. So a honeybee colony in a single honeybee colony in an environment at a 5-kilometer radius, it's going to do fine for the most part. But if you have like 1,000 of them at a given time in space and time, they're going to be competing for resources. They're not going to get enough for what they need. It's like having one pie of pizza for this entire room to split up. We're not going to have enough food. So this environment is going to have a carrying capacity, which is why beekeepers will constantly and frequently supplement the colonies with artificial pollen substitutes or resources. So we have this area of summer dearth where you have no resources but a lot of bee colonies in an area, and it's always a struggle. And then I moved out to Mississippi. Delta region is a mecca for row crop agriculture. It hasn't been studied. It's been studied, but in the context of pollinators, beekeepers generally tend to avoid it because of, one, pesticides in the area, and two, there are conflict of interest with the other stakeholders such as farmers and growers. So beekeepers are going to want more wildflowers out there and pollinator-friendly plants. They're not going to want pesticides being used, but in agriculture you need pesticides to be used to control pests, and also you need to maximize the amount of land that you have for your cash crop. So finding common ground between these two groups is very difficult. However, it presents a unique opportunity because the bloom time of these major crops, such as cotton and soybean, occurred during a summer resource dearth. So especially when I was in Texas, but all over the country, during the summer months, there is a huge resource dearth for pollinators until like goldenrod starts blooming again in the fall. But when I moved to Mississippi, I realized this isn't really the case. The colonies are still building up, and they're storing a bunch of honey, and that's because the major bloom times of a lot of the crops are around July, August for soybean and cotton. So in the country, we have almost 12 million or 11 million acres of cotton and almost 88 million acres of soybeans. However, you do come into the flip side of the coin where you have pesticides being applied in the environment. So you have to consider that to see if it's worth it. In terms of whether or not it's worth it for the farmers, the major crops are usually soybeans and cotton, and a huge reason why it hasn't been studied very much in the context of pollinators is because these two plants are commonly known to not need animal pollination. They're self-fertilizing plants. So they're like, why do we need pollinators if they are going to pollinate themselves anyways? However, a few studies have found that, and these are actually both reviews, that pollinators can actually increase the yield of soybeans and cotton. So there can actually be benefits to the farmers as well, in addition to beekeepers possibly using this area as a resource for summer dearth. So we did a huge review where we compiled a lot of information on what the nutritional quality of these resources, these different crops provide for pollinators. So we looked at both specialty crops and also principal crops. And one thing that we noticed during the search was we have a lot of information for specialty crops because a lot of them are known to be bee pollinated. So they need bee pollination. So that research is a lot of it's already done. So you have like citrus, for example, broccoli, which is known as brassica. Those are all bee-friendly plants that people have already looked into the nutritional properties of pollen nectar. However, for those principal crops such as cotton, even sweet potatoes, soybeans, peanuts, a lot of that, whereas pollinator-independent plants, they don't really have much information at all. There's like knowledge gaps on what the PL ratios are for protein and lipid ratios are for those plant resources. And so we are looking at this at kind of a complicated angle. We are looking at generally what the resources provide to bees and nutritional content there, but we are looking at things from like a bottom-up point of view. So have you heard of the saying, you are what you eat? Yeah. Your diet influences, yeah. A lot of your physiology and also nutrition. Well, the same thing can kind of be said for pollinators and also plants. So this is kind of an umbrella of, I don't even know how many now, maybe like 5 or 6 different studies where we looked at how different agricultural management practices, such as tilling and cover cropping, can affect the soil structure, soil health, nutrients in the soil, microbial activity in the soil, enzyme activity in the soil, and how that can affect the plants themselves and the physiology of those plants, and how that will ultimately affect the resources that those plants produce in the context and with implications of pollinator health. So in case you're not familiar with tilling and cover cropping, tilling is the practice of agitating the soil, turning it over, and it's very commonly done in conventional agriculture to control for weeds and prepare for seeding. But it does, over time, compromise soil structure and integrity, and soil health will just shoot downwards from there. So there are no-till management conservation strategies that can be done to minimize these effects of erosions, which is soil loss, nutrient loss, and also surface water runoff. And then there's a practice of cover cropping, which is the idea of planting these non-cash crops outside of the season, such as crimson clover and hairy vetch, or even rye, on agricultural land. And the idea is to help minimize soil erosion outside of the season, because when the cotton and soybeans aren't growing, you just have a giant landscape of dirt, and it's very prone to erosion. These cover crops, especially crimson clover, are known to be nitrogen fixers, so they can add nutrients back into the soil. It can promote overall soil structure as well and minimize soil erosion. So we did this study with Auburn University. Well, there are a few studies we did within the USDA, but the one I'm talking about here is one with Auburn, where we had 3 different treatment groups, and we wanted to look at how conventional conservation and halted management practices affected cotton and the resources they produce in the context of pollinators. So the conventional one was basically managed. Typically, they were tilled. There were no cover crops on there. Typical large-scale agricultural system. The conservation plot was planted with a cover crop using a drill, and then the halted treatment group was managed until the conventional treatment group. So same thing, they were tilled back then, up until 2018. So that we did the sampling in 2023. So it's been 5 years since the conventional management practices of tilling has been stopped. So we have these 3 treatment groups here. And these are just some images of bumblebees in cotton, seed pollinators in the cotton, potentially pollinating, and then us sampling the cotton flowers, bringing them back to the lab, and then digging up soil cores for analyses. So what we found with this, starting off with the soil, the enzyme activity was actually significantly different in the conservation plots, but it was significantly higher in these conservation crop management practice plots compared to the halted and conventional plots. So we looked at biohydrolase, which it helps break down cellulose, among other enzymes that helps degrades and breaks down other nutrients such as phosphorus and phosphatase, beta-glucosidase. And we also looked at microbial enzyme activity because the microbes in the soil can also help release nutrients. And we saw very consistent results throughout, where the treatment groups that had cover crops had higher enzyme activity, so you can suspect that the nutrient levels in the soil are going to be higher. So once the cover crops are planted and once they are done, they are terminated, and the organic biomass is left on the soil for it to degrade and break down, which is probably why you see all these enzyme activities much higher than the other 2 treatment groups. It is unfortunate that we don't see much of a difference. Oh, I forgot the letters here. But yeah, you don't see the difference between the halted and conventional groups, because that just to me it means that there are long-lasting impacts and effects of these conventional management practices that take more than these 5 years for you to kind of recover back to its natural levels. We also have data on the soil nutrients already, and the conservation plots, I don't have it here ready to show you, but I did make a PCA. But the conservation plots consistently had higher levels of nutrients compared to the halted and also the conventional treatment groups. So here's the 0 to 5 centimeter layer of the topsoil of the soil nutrients, and here's the 5 to 15. Once we get deeper, we don't see much of a difference here, but you can see that the PCA group and the data for the conservation treatment groups, the soil there is different, and in this case higher compared to the conventional and the halted groups. And how does this translate to nutrient content in pollen? Unfortunately, or actually I don't know if unfortunately is the right word for this, but we do see significantly lower protein content in the pollen in the conservation group, management group for cotton. So that's interesting because you have these higher enzyme activities and higher nutrient levels in the soil of cotton, but it translated to lower pollen protein content levels. There wasn't a difference in the halted and conventional, and also we didn't have enough sample size for lipids, but you can see a similar trend as well. And there are no differences for the nectar. However, we saw cotton actually has 2 nectaries. We saw a difference between the sugar concentration between the 2 nectaries. So there's one nectary, the one outside of the plant, the extra floral nectary. That one's used to attract ants to defend against aphids and other predators or herbivores. And then you have the floral, the nectaries inside of the flower are used to attract pollinators. And there was a difference there, but that's already been established. So to recap this part of the study, The conservation management practices did help promote soil structure for plants and increased soil enzyme and microbial activities that are involved with breaking down organic matter, which release nutrients in the soil and increase overall nutrition in the soil. But the change in soil structure didn't necessarily on protein content, and it resulted in reduced pollen protein content overall. But that doesn't necessarily mean it's a bad thing, because as we saw in the bee nutrition part of the presentation, bees require a balanced ratio of protein and lipids. Higher doesn't mean better for bees, and different pollinators actually have different nutritional niches. So some bees will prefer, such as bumblebees, who are the ones that are foraging on cotton the most, they prefer higher protein pollen. But other bees, such as Melisodes or Osmia, they may prefer a different ratio of protein lipids. So it doesn't necessarily mean that it is not a great thing for bees, the lower protein content. It might be better for different groups of bees. So just to recap and summarize everything we talked about today. So the pollen nutritional landscape can vary at spatial and temporal scale, and we found that honeybees actually collect different resources, and the nurse bees regulate their nutritional intake to about 1.4 to 1 protein lipid, which is consistent with a lot of the plants that they forage on. And then we saw that when we applied and introduced a pesticide, nutrition, possibly phytochemicals, can actually help upregulate the immune genes. So this is our suspicion. We haven't tested this